Yeah, so the 3 clinical trials that I discussed were all phase 1/2 studies investigating novel therapeutics in melanoma. The first clinical trial was by Merck. It was an umbrella trial looking at 3 different arms of an umbrella study. The first one was evaluating triplet therapy, anti-PD-1, Quavonlimab, targeting anti-CTLA-4, and then a TIGIT molecule. The second arm was also looking at that same PD-1 CTLA-4 combination with Lenvatinib as a TKI. And the third arm was looking at combination Pembrolizumab with ATRA. And unfortunately, what we saw is all 3 of the arms did not meet their pre-specified objective response rates to move forward with further enrollment. However, I think we did have a lot of takeaways, such as triplet therapies, looking at the tolerance certainly did add more toxicity without adding benefit, and I think a little lessons learned there in the field. The second study was with brenetafusp. So this is a bispecific, it's an ImmTAC molecule targeting PRAME. There are several molecules all targeting PRAME in the early phase setting. This trial was presenting on data in cutaneous melanoma. Certainly a lot of excitement there because of the diffuse expression of PRAME. So they evaluated monotherapy in patients with refractory cutaneous melanoma. While the objective response rate was 11% for the entire population and 13% for the PRAME positive cohort, there were certainly a number of patients that had very durable responses. This was evaluated in monotherapy, and they do plan to move forward in combination in the frontline setting. And this is a molecule that has had a weekly regimen that's been a little challenging just from a patient standpoint, but they are modifying that schedule in the frontline setting. So I think certainly some interest in just a different technology. An ImmTAC molecule has already been approved in melanoma, in the uveal melanoma space, so certainly a track record there, and I think some excitement moving forward. The third molecule was SC-682. This was a CXCR1/2 small molecule inhibitor. This was also explored in the early phase setting, so they looked at a number of different doses. We're really looking at toxicity. And what they saw is that there were a number of grade 3 events, but most of those were cytopenias, as you would expect from its effect on neutrophils. But there was no increase in infections or really the outcomes that we're concerned about for patients. It demonstrated a 21% objective response rate, which was very encouraging at this kind of early stage in the trial.